Welcome to Lecture 12, Part 1. This is going to be an overview of nutrition and metabolism, and we'll talk a little bit about carbohydrates, fats. In the next part, we'll talk about proteins, and we'll finish up with vitamins, minerals, and some miscellaneous topics in nutrition and metabolism. This is from Chapter 24 in Marib. First thing we want to define is nutrients. Nutrients, as you see here, are chemical substances supplied by the environment that we need for survival. In other words, these are things that the body doesn't make, so we have to get these from outside the body. And we can divide nutrients into three different classes: macronutrients, micronutrients, and essential nutrients. Macronutrients are those things that we have to take in bulk, things like carbohydrates, proteins, fats. Micronutrients, on the other hand, are things like vitamins and minerals that we only need very small amounts of, but these are very important for the body's function. And finally, we have essential nutrients. These are things that cannot be synthesized in the human body. We have essential fatty acids, we have essential amino acids, and of course vitamins and minerals, as we saw over here. So these are the three different classes of nutrients that we have. Now what I want to do is just do an overview of the general metabolic pathways that we have. Since these will be important, we're going to be talking about each of these major macromolecules: lipids, carbohydrates, and proteins. So let's define the general metabolic pathways. This one you'll be most familiar with, that is the breakdown of carbohydrates to generate ATP in the mitochondria. And we talked about this in Anatomy and Physiology 1. Remember, there are three major sections to this. We have the breakdown of glucose into pyruvic acid. This whole process was known as glycolysis. This generates pyruvic acid, which is decarboxylated to acetyl-CoA, which is here. Acetyl-CoA is a 2-carbon molecule, whereas pyruvic acid is a 3-carbon, and this is where we lose that 1 carbon in carbon dioxide that we exhale. The acetyl-CoA goes into the TCA cycle, and very importantly, as we'll see later, it merges with a molecule called oxaloacetate, or oxaloacetic acid, and we'll cover the metabolism of that a little while later. Acetyl-CoA goes to the TCA cycle. Remember, this is also called the Krebs cycle, or the citric acid cycle, and the products of this are more CO2, more electrons, and more ATP. Now all the electrons that are generated in glycolysis, the transition of pyruvic acid to acetyl-CoA, as well as through the TCA cycle, are funneled, if you remember, into the electron transport chain, which needs oxygen. So we say this is an aerobic process, and the electron transport system uses those electrons, harvests the energy from those, and generates most of the ATP that we have in the cell. An additional byproduct of the electron transport system is water. Now you notice the interconnection of glycolysis and this main pathway with some other things. The overall goal of catabolism, and really generally metabolism in the body, is to generate a pool of 2 or 3 carbon molecules as well as ATP. The 2 and 3 carbon molecules we're talking about are right here. Pyruvic acid, if you remember, has 3 carbons. Acetyl-CoA, as we just mentioned a second ago, has 2 carbons. And so we lost 1 carbon here in carbon dioxide. These are what we call the 2 and 3 carbon intermediates of metabolism. Very important because these can be used to synthesize a number of things. Let's take a look first at pyruvate. You'll see that pyruvic acid is used to synthesize amino acids as well as acetyl-CoA. So we already know that pyruvic acid can be converted to acetyl-CoA in this step on the way to generating ATP. However, pyruvic acid can also be used to generate amino acids, as you see here, indicated by the blue arrow. The arrows on this diagram indicate whether we're talking about catabolism or anabolism, depending on their color. Anabolic pathways are labeled with blue arrows, catabolic pathways are labeled with red arrows. So you see the conversion of pyruvic acid to amino acids on the way to producing proteins is an anabolic type process. Pyruvic acid can also be used to synthesize glucose. This is essentially the reverse of glycolysis, and this is known as gluconeogenesis. Now if we break that word apart, and we'll talk about this a little later, gluconeogenesis, glucose sugar, neo, new, genesis, creation of. So literally this is the creation of new sugar. We take things like pyruvic acid and some other things like oxaloacetate, and we can put them in this process called gluconeogenesis to generate glucose. Now, what is the fate of glucose in the cell? Well, glucose, when we have it in excess, and it doesn't have to be used to generate ATP, can be used to generate glycogen. Remember, glycogen is the storage form of sugar that we use in the human body. This is generally stored in the skeletal muscle as well as the liver. This process of taking glucose and making glycogen from it is known as glycogenesis. Once again, we have that word genesis, which means creation of, glyco meaning sugar. So this is our storage form of glucose, and this is the process of glycogenesis. We can also go the other way. When we need glucose in the body, we can take the stored glycogen and break it down by a process known as glycogenolysis. So glycogen, olysis, breakdown. We're breaking down glycogen, we can generate glucose, and we can use that for several different things. Now the last pathway here that I want to talk about is the pathway that connects the carbohydrate metabolism to lipids. And if you look down here at the arrow for glycolysis, you'll notice that we can have glycerol come out of the glycolytic pathway. And remember that glycerol is one ingredient that we need to make triglycerides. The other ingredient were fatty acids. So we need 3 fatty acids plus a molecule of glycerol, and this will create triglycerides or fats. And we call this process of creating new fats lipogenesis. Lipofat, genesis, creation of. We can also go the other way. When we don't have enough sugar in the body and we need to turn to fats for energy, we can break down those stored triglycerides into fatty acids and glycerol through the process of lipolysis. And you'll notice that the way this is connected to the main pathway that we looked at before is through this molecule acetyl-CoA. So acetyl-CoA can be used anabolically to create new fatty acids which can be used in lipogenesis. On the other hand, we can break down fatty acids and generate acetyl-CoA that can go into this pathway and then be used to make energy. One thing I want to remark on here is the arrow that you see going from pyruvate to acetyl-CoA. This is a one-way process. In other words, we can't go backwards from acetyl-CoA to pyruvic acid in humans. So acetyl-CoA has its own routes of metabolism. Pyruvic acid has its own routes of metabolism, but the acetyl-CoA cannot go back into pyruvic acid. So I hope this refreshes your memory a little bit and introduces some things that you may not have known about the connections between these pathways. Keep in mind as we talk about the things that we're going to discuss in nutrition, the importance of these very central intermediates of metabolism in transitioning from one metabolic substance to another. Another important point here that I want to make before we go is that we can take carbohydrates, and from this we can make lipids, or we can make proteins. We can take proteins, from these we can get energy. We can also eventually take proteins and we can make lipids. We can also take lipids, we can put it into this carbohydrate pathway. So you see that we can transition between these different kind of metabolites, and the main reason we can do that is because of these small 3 and 2 carbon chains that we have here. Okay, so let's talk about the first major class of macromolecule we want to discuss, and those are carbohydrates. Remember, these are also called saccharides, as we said in AMP1. Several of the sources of carbohydrates for us include simple sugars, like sucrose, for example, common table sugar. Some of the other sugars that we take in, malt sugar, for example, lactose, milk sugar. Starches as well. These are starches from things like potatoes, pasta, wheat, and those sort of things, mainly from plant sources. We can also, of course, take in starches from animals in the form of glycogen. For example, when we eat muscle meats, that's glycogen that we're taking in, which is a complex carbohydrate. And finally, remember that plants have a form of polysaccharide known as cellulose, which forms for us indigestible fiber. In other words, we can't break down cellulose in our own digestive systems, and this forms a very important substance in our body that is indigestible fiber that helps to cleanse the colon and also helps retain water in the feces so that constipation doesn't occur. We talked about that in connection with the digestive system lectures. Now, the recommended daily allowance, as you see on the bottom, for carbohydrates is about 125 to 175 grams per day. Now, the recommended dietary or daily allowance is a number that's established by our government just as a general guide to the minimum amount of something that will ensure good health. Although we can criticize our government for a lot of things, we really have to give them credit where credit is due here. To make a minimum recommendation for the entire population is very, very difficult. To come up with something that benefits everybody as a whole is very, very difficult. This is considered a general guideline for the major population, that is, people who are not sick, not pregnant, not nursing, not athletes. So there are many different things that this doesn't really take into account. And those things have to be adjusted, for example, by dietitians, and people who know a little bit more about nutrition will have to amend these guidelines so that proper nutrition is maintained for those segments of the population who need a little bit more or even a little bit less than the dietary or daily allowance. Okay, now excesses of carbohydrates, as we probably know, can lead to obesity and dental caries, which are cavities. This is not very hard to understand when you think about the fact that on average in America we consume about 100 pounds of sugar every year. 60 pounds of that in the form of sucrose, or common table sugar, the other 40 pounds in the form of corn syrup. And the corn syrup is one of the things that's really gotten a bad rap, justifiably so. Very, very bad for us. The only problem is it's pretty much in everything that we eat. We find it all over the place, and it's very hard to avoid corn syrup. So this is one of the reasons why we find the population of the United States so obese, and in spite of the advances in dentistry, we find so many people developing dental caries or cavities. The other thing that can happen is an excess of anything can lead to nutritional deficits. That's the case as well with carbohydrates. If we take anything in excess, that can basically leave out the other things that are important in our diet as well. So we say that we need a balanced diet, and that means we have to include some of a lot of different things. Now, deficiencies of carbohydrates can lead to something called metabolic acidosis. We'll talk about this a little bit later. And deficiencies can also lead to weight loss. One of the things people do when they go on diets is to restrict their carbohydrate intake. And of course, there are different kinds of carbohydrates. As we said, there are sugars, which generally are bad for us and lead to things like obesity. On the other hand, there are starches, glycogen, those sort of things that are better for us. And complex carbohydrates from plant material is much better for us than the simple sugars. Now, how do we use carbohydrates in the body? Well, we take carbohydrates into the body in different forms. For example, starch, as we said, sucrose, common table sugar, maltose, malt sugar, lactose, milk sugar. And you notice that through the process of digestion, what we do is we break these down into the common denominator, that is glucose. Glucose is, as it says at the bottom here, the main source of energy for the body. Now, we can use the glucose for several different processes. Through the process of cellular respiration, we can generate energy in the form of ATP. And as we know, this also generates carbon dioxide and water. If there's excess glucose, we can store that excess glucose as glycogen through the process of glycogenesis. And finally, excess carbohydrate can also be stored as fat through the process of lipogenesis. And we looked at some of the connections between these pathways on the previous slides. Now, what about lipids? Let's turn our attention there. Several sources of lipids. You'll notice meats, eggs, milk, lard, plant oils. This, of course, is a highly saturated fat and is one of the things that most people stay away from these days. Butter, milk, eggs, these things contain different kinds of fats as well. Meats also contain a high proportion of saturated fats, so we have to limit our intake of meats and preferentially eat those meats that are a little bit lower in fat. Plant oils are a great source of lipids or fats because many of these plant oils contain unsaturated fatty acids. Now, the recommended daily allowance is about 80 to 100 grams of lipid every day. These are very important guidelines, this one especially. We should limit our intake of calories from fat to no more than 30% of our total calories. In other words, if we have somebody that has a 2,000 calorie diet, 2,000 calories per day times 30%, that would be 600 calories per day maximum from fat. So 30% of calories is the maximum that we should be taking in from fat. And in fact, only about 10% of the fat that we take in should be of the saturated variety. As I said, the things like meats, milk, butter, lard, those sort of things have a higher proportion of saturated fat, so we have to limit our intake of those sort of things. Excesses of lipids, as you probably know, can lead to obesity directly by the triglycerides that we take in, the other fats being broken down and then resynthesized into lipids that are stored in adipose tissue. And very importantly, excesses of lipids, especially saturated fatty acids, can increase serum cholesterol, and this goes hand in hand. This can increase the risk of heart disease. Now, some of the effects of deficiencies of lipids include weight loss. Many people go on a diet, and one of the first things that they restrict is the amount of fat that they take in. However, if we restrict fat too much, what we can wind up developing are skin lesions and hormonal imbalances. These are very bad things. We have fats in the skin that are very important to keep the skin as a protective barrier. We talked about those in the integumentary system in AMP1. For example, we talked about lamellar granules. Those are fat-based molecules that help the skin remain water resistant. Some hormones, like steroids, use cholesterol as their base. So any kind of deficiency in cholesterol can result in hormonal imbalances. Now, fats usually take longer to digest than the other macromolecules like carbohydrates or proteins. Why would that be? Well, think back to the digestive system lectures and remember that we had that molecule cholecystokinin, my favorite hormone of digestion, and it turns out that the more fat that enters the duodenum, the more cholecystokinin is secreted. If you remember, cholecystokinin inhibited stomach emptying. Therefore, the more fat in the meal, the slower the stomach will empty. That keeps us full for a longer time. So this is the reason why fats generally take longer to digest or get through the alimentary canal than other molecules. Now, how do we utilize lipids? Up in the left-hand corner of this slide, you'll see the structure of a triglyceride. Remember, we have a glycerol connected to 3 fatty acids. So if you look at this diagram, you'll see on the bottom right of this slide that we have a key, and basically we have different color arrows on this diagram that are indicating whether something is lipogenic or lipolytic. In other words, whether we're going to make new fats or whether we're going to break down fats. The red arrows are indicating lipogenesis. If we were to take in an excess of glucose, you can see that through the glycolytic pathway, we can generate glycerol. We could also generate acetyl-CoA from that same glucose molecule. These two things together will generate new triglycerides, and that is the process of lipogenesis. The reverse, as we said, is also true. We can have stored triglycerides that can be broken down into glycerol plus fatty acids, and glycerol can go into the glycolytic pathway. The fatty acids can undergo what's called beta oxidation, which we'll look at in a second. This can generate acetyl groups and eventually generate acetyl-CoA that can be funneled into the citric acid cycle to make ATP and go a couple of different directions in the body. Let's take a look at the bottom of the slide, and you notice a molecule I mentioned before, which is called oxaloacetic acid, or sometimes oxaloacetate. This is what's called a pickup molecule for acetyl-CoA. This is a 4-carbon molecule. This is a 2-carbon molecule, as we talked about before. When these two combine, they make a 6-carbon molecule, which is called citrate, and hence the name citric acid cycle. Very importantly, acetyl-CoA, in order to be funneled into the citric acid cycle, or the TCA cycle, to generate ATP, has got to have oxaloacetate to merge with it. When there's not enough oxaloacetate in the body, the acetyl-CoA cannot go into the citric acid cycle. Instead, it's funneled a different direction. And it goes to create some of these things that are known as ketone bodies that you see circled on the lower left of this slide. Ketone bodies such as this include beta-hydroxybutyric acid, acetoacetic acid, and acetone. Ladies, many of you will know acetone because this is the main substance in nail polish remover. So this is the smell that you smell when you use nail polish remover. Now these things are acidic bodies. They make the body more acidic, as you can see from the word acid after these two substances. Acetone is a very volatile substance that can be breathed out. So in many diabetics who don't have enough oxaloacetic acid because they can't use carbohydrates very well, what happens is they don't have enough oxaloacetate to merge with the acetyl-CoA to make ATP. So instead, what happens is the acetyl-CoA is funneled into making ketone bodies, and the ketone bodies then produce ketoacidosis because of the acids here. In a severe case of diabetes or in somebody that has highly elevated blood sugar, one of the things you can often smell is this kind of fruity odor on their breath, which is produced by the acetone from these ketone bodies. So acetyl-CoA and acetyl groups are really at the heart of all of the production of ketone bodies. Now, a couple of questions just to refresh your memories. Via what structures is fat absorbed into the circulation from the digestive tract? And if you remember, there were structures called lacteals that were present in the small intestine that absorbed fat in the form of chylomicrons. And these, remember, bypassed the portal circulation. They went directly back to the heart, and then on the second pass, they'll wind up going to the liver. The terms on the bottom of the slide here, lipogenesis, lipolysis, these were on pretty much the very first metabolic pathway chart I showed you. These are important for you to know. Lipogenesis, creation of new fat. Lipolysis, breakdown of existing fat. Now let me talk a little bit about the beta oxidation of fatty acids. This is a fatty acid, and you'll notice that this is an 18-carbon fatty acid, and these are long hydrocarbon chains as you see here. And what happens in the metabolism of fatty acids is that the carbons are cleaved off fatty acids 2 at a time. So you basically see this one unit here on the end. These 2 carbons will come away from the fatty acid and make acetyl-CoA. And so acetyl-CoA from this fatty acid can go into the TCA cycle, as we mentioned before. We can make acetyl groups here. Remember that these acetyl groups not only can go into acetyl-CoA into the TCA cycle, but they can also go to make those ketone bodies in the case where we don't have enough oxaloacetic acid to merge acetyl-CoA with in the TCA cycle. So this is what beta oxidation of fatty acids is. Now, long-chain fatty acids like this one, like the 18-carbon fatty acid we're looking at here, require something called carnitine for transport into mitochondria so that the beta oxidation of fatty acids can be carried out. As you see, this is a process that occurs in the mitochondria of the cell. Fatty acids are broken down in a 2-carbon stepwise manner, as we just said, and this is the process of beta oxidation. And finally, oxidation of fatty acids yields about 1.5 to 2 times the energy from a comparable amount of glucose. Now we're going to talk about the number of calories per gram of different macromolecule in a second or two. So let's turn our attention to the energy values of food. These are the things that you would see, for example, on a label in a store. And when we talk about calories in food, we're really talking about something that's known as a big calorie and what's really called a kilocalorie in scientific terms. This is 1,000 times the typical calories that we're talking about when we, for example, do chemistry reactions. So remember, a calorie is the amount of heat needed to raise the temperature of 1 gram of water by 1 degree centigrade. A big calorie, with a big C, is the amount of heat needed to raise the temperature of 1 kilogram of water by 1 degree centigrade. So we're talking about 1,000 times the energy in those small calories that we talk about in chemistry. So in nutrition, when we talk about carbohydrates, we say that they have about 4 calories per gram. Proteins yield about 4 calories of energy per gram. Lipids, on the other hand, because these are so nutrient dense, yield about 9 to 9.5 calories per gram. You can round these off if you want for the purposes of the exam to 4, 4, and 9, just to make your calculations a little bit easier. But you do need to know these numbers. Now let's just do a couple of sample calculations. A diet is 2,000 calories. What is the maximum number of grams of fat that should be taken in daily? 2,000 calories per day. That's the number of calories that we're taking in per day. And we know from this that we have a conversion factor to convert calories to grams, right? 9.5 calories per gram. Remember, if we want to get rid of units, we have to put those on the bottom. So we say 9 calories down here in 1 gram of fat, and that's going to yield a total of about 222 grams of fat per day. So if our diet was purely fat, we could take in 220 grams per day, and that would be the equivalent of 2,000 calories. But do we actually want to take in 222 grams of fat per day? No. If you remember, we talked about that. It's much less. And in fact, we only want to take in 30% of that. So if we take that 222 grams per day and we multiply that by 30%, we get a figure of about 67 grams of fat per day. So this is the maximum number of grams of fat that we want to take in per day based on a diet that's about 2,000 calories. Another question for you. How many grams of fat do you think there are in a Whopper with cheese and a small order of French fries? Now, I'm not picking on Burger King. There are many fast food places that have similar type hamburgers and side orders and those sort of things. But it might surprise you to know that the number of grams of fat that we have in a Whopper with cheese and a small fry are about 59 grams. And as we just looked at, if you have a 2,000 calorie diet, the maximum number of grams of fat that you want to take in are about 67. So this is very close to the maximum per day. So in one meal from a fast food restaurant, if you're not careful, with cheese, with mayo, having a side of French fries, and perhaps something else, this can raise the number of calories dramatically. Let's take a look at this label that we see. This is similar to the nutrition labels that you'll see on different foods in the store. And let's use our newfound knowledge in terms of the calories per gram to look at this. You'll notice that the carbohydrate is given here. The protein is given here. So what we want to do is calculate from that how many calories are contributed to this total calorie intake from this beverage, which happens to be beer, one of my favorite food groups. We want to calculate total number of calories here. Notice that we have 14 grams of carbohydrate, and that is 4 calories per gram for 56 calories. We only have 1 gram of protein times 4 calories per gram. That's 4 calories. That makes a total of 60 calories. But wait a minute. If you take a look up here, notice that the total calories in this one bottle of beer is 166. So why in this 12-ounce bottle of beer do we seem to have more calories than what are listed on the label? Well, as you might guess, the answer is alcohol. Alcohol has about 7 calories per gram, almost as much as fats do. So alcohol is really a very high calorie substance. One of the reasons people gain weight, especially during holidays and those sort of things, when people drink a little bit more, they don't realize how many extra calories they're taking in just from alcohol. So something to be aware of if you're looking to lose weight or if you're looking to maintain your weight and don't want to gain any weight. Keep in mind that alcohol definitely has calories. Now let's talk a little bit about lipoproteins. Lipoproteins are fats that are surrounded by a protein shell, so we call them lipoproteins. And we have different classes of lipoproteins that I want you to be familiar with. The liver is the main site of processing and transformation of lipoproteins in the body. Let's follow along with the numbers on the diagram. Here's number 1, and what the liver does with the dietary fat that comes from the small intestines and eventually comes up through the lacteals and through the lymphatic system. It first creates VLDLs, which are called very low density lipoproteins. Now, the density of a lipoprotein is really dependent on the ratio of protein and fat in the lipoprotein. The more protein there is, the more dense. The more fat there is, the less dense. So a very low density lipoprotein is going to be carrying a lot of fat. And in fact, these are triglyceride-rich type molecules. Triglycerides are very important for the body, and especially fatty acids, for use by cells. So the liver makes very low density lipoproteins. These triglycerides that are being transported are broken down in the capillaries near the tissues, and they'll liberate fatty acids and monoglycerides for use by tissue cells. Now, the remnant of the VLDL that's left after it unloads the fat is known as an IDL, which is an intermediate density lipoprotein. Shouldn't surprise you, because we have a protein coat here, and we've taken out some of the lipid from the inside, and so now this molecule has a higher ratio of protein to fat than a VLDL did, and consequently we call these intermediate density lipoproteins. These go back to the liver. As you'll see at number 3, what happens next is the liver repackages these, takes some cholesterol from the diet, and will make these particles that are known as LDLs. These are low density lipoproteins. So these are a little bit more dense than the VLDLs we looked at before, and these primarily carry cholesterol. So these are cholesterol-rich molecules. Cholesterol, as we talked about a couple of times in the course, is important in, for example, the synthesis of sex hormones, some other hormones, in the synthesis of cell membranes. So the cells do need a minimum amount of cholesterol to do what they need to do. The LDLs deliver cholesterol to body cells. This is taken up by the peripheral cells, broken down in the lysosomes, and the cholesterol that comes from the LDLs is used by the cell. Any excess is transported out of the cells and picked up by molecules that are known as HDLs, high-density lipoproteins. These have a relatively low percentage of fat to protein, so these are higher density. And these are transported, as you see, back to the liver to be metabolized again. The cholesterol, whatever cholesterol they have in them, is used to repackage into LDLs again. So there's a circulation of LDLs. The other thing I want to call your attention to here is that cholesterol can be manufactured from acetyl-CoA in the liver. I don't know if you've ever heard those drug commercials, but we actually do have 2 sources of cholesterol, from our diet and from our own bodies. That is, from our genetics. So our body does manufacture cholesterol from acetyl-CoA. This is a summary table of lipoproteins. So these are the important facts for you to know. I'd like you to know the entire table, the designation of the different lipoproteins, their origins, where they come from, and then the actions of these, what they do. Finally, let's talk a little bit about the utilization of lipids. The liver uses fatty acids to synthesize a whole variety of lipids, as you see on the bottom. So the liver can manufacture triglycerides that will be stored in adipose, lipoproteins, as we just saw, that can transport different fats around the body. Fatty acids, except the essential ones that we're going to mention in a second. And these fatty acids are used for the metabolism in cells. They also make, of course, cholesterol, phospholipids that are used in cell membranes. So the liver is very versatile in terms of its synthesis of a whole variety of lipids. And as we said before, we have the pathway of triglycerides that undergo digestion. Eventually, these fatty acids and glycerol are passed to the liver through the chylomicrons, and then this does its magic. Now, there are a couple of things called essential fatty acids that I want to take a look at. 2 essential fatty acids that we need as humans include linoleic, which is called an omega-6 essential fatty acid, and linolenic acid, eicosapentaenoic acid, docosahexaenoic acid. These are all what are called omega-3 type fats. These are believed to be more beneficial than the omega-6. And in fact, really, it's the ratio of omega-6 to omega-3 that's important. We need to have more omega-3s in our diet than omega-6. We can get the omega-3 oils like EPA, DHA from fish, so many people take fish oil. Although that can be a problem as well, because in some of the larger fish that eat the smaller fish, the fish oils have higher concentrations of mercury. So this can be dangerous if the fish oils are not processed correctly. So I recommend if you do take fish oils that you look for some that are molecularly distilled so that the mercury is left behind and just the good fish oils are there. Now, where do we get these designations omega-6, omega-3? If you look at the structure of the fatty acids up here, you'll notice that the opposite end of these fatty acids from the carboxylic acid end is designated as omega. Sometimes people call this N, but we'll use omega. And you notice that the number of carbons that we have before we have the first double bond. So here we have carbon 1, carbon 2, carbon 3, and you notice that this is the appearance of the first double bond, so this would be called an omega-3 type oil. This one over here has its first double bond at carbon number 6, so this would be known as an omega-6 type oil. And as I said before, really the ratio of omega-6 to omega-3 fatty acids in our diet is important. It's recommended that we take in more omega-3 oils than omega-6. Typically in our American diet, though, we tend to have more omega-6 to omega-3. That's one of the reasons why people take fish oils. Okay, that does it for part 1 of our nutrition lectures. I'll see you very soon for part 2.